Very good evening to all of you present over here. Please confirm if I am loud and clear and if you are able to see the slides. Okay. So let's get started with today's session. So I'm sure you all are excited today so we have come to the day 6 of our session, right? So it is, as you all know, we have started it from 28 June and we'll be continuing it till 13 July. And I'm sure you all are using the maximum benefit of this sessions that are going on. So today I am Dr. Violet and I'll be taking up a topic from Unit 3, that is Biology and Human Welfare. So the topic from this unit that I'll be today discussing is basic concepts of immunology. Along with it, we are going to also talk about vaccines. Now, I'm sure you all are excited about this topic because it is a very interesting topic, right? We all, presently in the present scenario, we all might have, read so many news articles related to our immune system, immunology, right? So this is one of the most important and interesting topic. And along with it, yes, vaccines. So today, like everybody is talking about vaccines, so even about vaccines we'll be discussing. So apart from being an interesting topic, it is also an important topic from your CUET exam. Okay, so you have to be well prepared with this particular topic. So let's get started. We will discuss all about immunology. Right now, before I start with the session, I request you all, whoever has already downloaded the app, BioTecNika app, it's well and good. Whoever has not downloaded, please download this app. You will be able to reap the maximum benefits that BioTecNika provides for its subscribers. Right, you can get access to various classes, videos. You will get to know about the various applications, the newsletters, the job opportunities. So it's a really no, you get everything in one app. So just download the app, and even you can be in touch with BioTecNika. For that, you need to download the Telegram app. You need to join the Telegram group. Okay, so the link is provided over here. So before we start, I'm sure you all have done this. So let's get started. Okay, so first, what is immunity? Right, immune system we have heard. Then what is this immunity? Okay, so immunity means whenever any pathogen enters our body, right? So our body will try to protect itself, right? So you can say that it is the overall ability of the host, that is the human we are talking about, the body, to fight the disease causing organisms. Okay, so it can be any of the pathogen, the various microorganisms. It can be a virus, it can be bacteria, right? So the ability of the body to fight those pathogens. Okay, so that is what is termed as immunity. So the ability of our body, that is the body has different systems, right? So in that, the immune system is the system that actually plays a role in protecting our body against the various pathogens. So that ability of the body or the immune system is known as immunity, by which it can fight the various pathogens, right? Now, this immunity is, again, you can broadly divide it into two types: innate immunity and adaptive immunity. Like, for example, what is innate? So you can say it is a first line of defense, right? So if you talk about the entry of the enemies on our border lines, right? So we have the army that is standing over there, right there, right? So you can say it is the first line of defense. So any infiltrators try to approach, so we have the first line of defense that is standing over there to protect our country. So same way, even our immune system, the first line of defense that is always standing right there to protect you against any pathogens that enter our body is known as the innate immunity. Okay, now suppose there is a breach in that innate immunity. okay, or the enemy is so strong, the pathogen is so strong, it cannot enter, means it cannot be controlled. Okay, so in that case, the next is the second line, you can say, of defense is the adaptive immune system, okay, or adaptive immunity. So first we'll talk about what is this innate immunity. So innate immunity, as I said, it is the first line of defense. Now what all comes in your mind when we talk about the innate immunity? Definitely, I'm sure you might be pondering about the various cells, right? Like we have neutrophils, we have basophils, so you can call them the various types of WBCs, right? So these play a role in protecting your body. So again, WBCs, like as I said, neutrophils, basophils, eosinophils. Then another one is your monocytes, right? So these are very important cells: monocytes, macrophages. Then we have, apart from that, we have the other cells, okay, like the WBCs. Then there are mast cells, right? So all these different types of cells will come under your innate immunity. Okay, so these are phagocytic in nature. They can perform phagocytosis, where if any pathogen is there, so they can extend their, the pseudopodia like structure, and they'll take, suppose it's a bacteria, so through this pseudopodia like structure, they'll engulf this bacteria. So this process is known as phagocytosis, which usually these cells can perform, right? So the response that is generated whenever the innate immunity gets activated, so the response gets generated within hours, means it is very quick. Okay, but there is one limitation, you can say, that it has limited specificity. Okay, limited specificity means, so it is just a first line of defense where, so we have different strains of bacteria, right? We have streptococcus, staphylococcus, so we have different strains. Okay, but when we say limited specificity means there is just one mechanism where the phagocytic cells will recognize some broad molecule that is present in the pathogen, maybe a LPS that is common to various bacteria. Okay, and then it will try to phagocytose. So here it cannot identify, it cannot distinguish between a bacteria 1 and a bacteria 2. Okay, so for it, both, for both of it, for both these strains, if they are belonging to different strains, so they have the same mechanism, so they show limited specificity, they're not highly specific. That is what we can say over here, right? But yes, why limited specificity means still some specificity shows. Why we are saying that it is showing some specificity? Because it can identify between a bacteria, and it can also identify between a virus, or it can identify with some other type of pathogen. Okay, so in that way it is a showing limited specificity. Now another important characteristic of innate immunity is it does not have memory cells, no memory. Okay, so when we talk about adaptive immunity, so there you will see that it has a memory response. So over here there is no memory response, means if the pathogen enters today, so the mechanism gets activated. Okay, it will try to control the pathogen. Suppose after 10 days again the pathogen enters your body, so the mechanism will again start from scratches. There is no memory cells that will be present, that will activate this response. Okay, so there is no memory cells. So subsequent response of identical intensity, so how much ever the primary response was there, so with the same timing, with the same intensity, the secondary response will also be there, even though the pathogen entered the body the second time. Okay, so all these are the characteristics of your innate immunity. Then what is adaptive immunity? Adaptive immunity is a second line of defense. So when this innate immunity fails, after even activating the macrophages, even controlling the bacteria, still the bacteria colonies are growing and they are not able to control the bacterial load, so the adaptive immune system gets activated. Okay, so the response generated within days, it takes little time. Okay, so the response is not quick as compared to innate immunity. So the cells that are involved in adaptive immunity are the B and T cells. Okay, so these are the lymphocytes. So these are also a type of WBCs. So the B lymphocytes and T lymphocytes are the ones that are associated with adaptive immunity. Diverse specificity. So what is this diverse specificity? So diverse specificity means here these B cells, T cells, they are capable of identifying the different, the different bacterial strains. So even if there is one amino acid change, Okay, so if bacteria 1 has one particular amino acid and that another bacteria has a different amino acid, so it can identify that, and accordingly the B cells will get activated. So there'll be one B cell. If I say B1 is specific for this strain 1, there'll be another B cell that is B2 that will be specific for another strain of bacteria, that is bacteria 2. Okay, so in this way it shows diverse specificity, right? Then memory, yes. So here you also see the memory response. So suppose the first time the pathogen entered, the B cells got activated. When the B cells get activated, what happens is they proliferate. Proliferate means there'll be a large number. Suppose a B1 got activated, so there'll be a large number of B1 cells that will be formed. Okay, then there'll be some signaling, so B cells will get activated. Okay, so when they get activated further, they'll differentiate. So when they differentiate, they form plasma cells and memory cells. Okay, so B cells differentiate. So you remember the terms, they differentiate and form plasma cells and memory cells. Plasma cells will release the antibodies, okay, that is required to control the pathogen, while memory cells will be there, the memory B cells will be there. And when there is a second exposure to the same pathogen, so quickly these memory cells will get activated. Okay, so that is what it means over here, that adaptive immune response shows memory. So same in case of T cells as well. So it improves with successive encounter with pathogen. Why it improves? Because there is the presence of these memory cells, right? Okay, now next, coming to what all comes under innate immunity. So first I told you there are different cells that are involved in innate immunity, right? Now apart from these cells, we have our physical barriers, the physiological barriers. Okay, the cells will come under the various cellular barriers. Then even there are cytokine barriers. I'm sure you might have heard of this term cytokine. If not, we are going to see over here what are these. So the physical barriers also come under innate immunity, like we have our skin, right? So the skin covers whole of the body. So what is the use of the skin? It protects your body. It does, if there is any cut in the skin, so what happens? The pathogen can enter through there, right? But overall, that is why the skin is present, so that the pathogens do not enter your body, right? So these are the physical barriers. Then apart from that, in the respiratory linings, in the gastrointestinal tract, in the urogenital tracts, okay, you have mucus coatings of the epithelial cells over there. So these mucous membranes, so there is also the presence of epithelial, the ciliated epithelial cells. So this mucus that is present, so even if a pathogen enters through your respiratory tract, so the mucus lining, through the movement of the cilia, will try to remove those pathogens. Okay, it will try to just sweep it off. Okay, so it cannot colonize. So in that way, it cannot cause infection, right? So that is the use of this physical barrier. Then cilia, as I already told you, what cilia does, it tries to remove any kind of pathogen, so it does not allow the colonization part. Okay, so these come under your physical barriers. Then what is a physiological barriers? Physiological barriers will include the pH of our body is such that many pathogens cannot thrive on it. Okay, like we have acid in the stomach, it is acidic in nature, right? There is HCl production in the stomach, so many pathogens cannot grow over there. Okay, then the saliva, okay, it has a particular pH where pathogens cannot survive, some many pathogens. Okay, so this comes under your physiological barriers. Apart from that, even a body temperature is such that many pathogens cannot thrive on it, they cannot colonize. Okay, they cannot grow in that temperature. Okay, so these come under the temperature, pH will come under the physiological barriers. So these are also a part of your innate immunity. Then the cellular barriers, so basically the polymorphonuclear leukocytes, like the different types, okay, where you have neutrophils, basophils, eosinophils, okay, and monocytes, okay, the NK cells, the natural killer cells in the body, I told you, mast cells. Then there are dendritic cells. So these are different types of cells that play a very important role, okay, that protects your body against the pathogens. So these cells, remember, come under your innate immunity. Okay, and how they do that? Definitely there are certain receptors. Okay, so there are receptors that are present on these cells, so they can identify certain chemical molecule that is present on the pathogen. So in that way, they can easily identify the pathogen. right? Now next is the cytokine barriers. So what is the cytokine barrier? Cytokines, okay, what are cytokines? Cytokines are very small molecular weight proteins, okay. They are basically involved in cell to cell communication, okay. And whenever there is any pathological conditions, okay, like if there is inflammation where there is a swelling in the body, so some cytokines are involved. Similarly, if there is allergic reaction, so some cytokines are released. So there are large number of cytokines, okay. Similarly, if your body is infected with a virus, so the type of cytokine that is released is known as interferons, okay. So this will protect the body against the viral growth or the viral infection. So it will protect the non-infected cells from viral infection. So remember, interferons are released whenever there is a virus entry, okay. So these cytokines are specific, like for inflammation or for swelling, you have some typical cytokines, like you have IL-1, okay, for inflammation. Similarly, TNF. Okay, so all these are involved in inflammation. IL-6. Okay, so these are, and then IFN gamma. So like that, there are various cytokines that have some specific functions. So these come under your cytokine barriers, which are again a part of your innate immunity. Now, further, if you see, okay, in innate immunity, what all comes? So there are various enzymes that are released in a body, like lysozyme is present in the tears. So this will also protect you against the pathogens because it tries to, break down the cell walls of the bacteria. Okay, so this also comes under lysozyme. Then skin, we already discussed. Now, another important aspect of skin is what happens is, you might have seen that the dead cells, okay, in our skin, it slows off, right? The whitish thing comes out. So the dried and dead skin slows off. So why that slowing happens? Because even if the pathogen tries to colonize, okay, it cannot colonize because the skin will come off, okay. So that is another way where it will inhibit the pathogen colonization, as well as the skin will produce certain antimicrobial fatty acids also, okay. So in that way, the microbes cannot thrive, okay. So we spoke about pH. Then the flushing of urinary tract. So when the urine moves, so it actually flushes. If any pathogen enters through the urogenital tract, so it cannot colonize. Then the various pathogens are removed through the function of cilia. I already discussed about this. Then the various blood proteins, okay, like various cytokines will also play a role over here. Mucus, cilia lining that are present in the trachea, so those will also protect against the entry of pathogens, okay. Then the acidity of the stomach, the normal flora also compete with pathogens. So in our intestine, there are some gut bacteria, right? So in this way, they don't allow the other bacteria to colonize, okay. So that is how the normal flora that is present in our body would compete with pathogens. So similarly, these also come under your physical and physiological barriers, the various enzymes, okay, and the flora part. Okay. Now, next is about the acquired immunity, okay, or you can also call it as adaptive immune response, right? So, adaptive immune response, as I said, it includes two types of cells, that is B cells and T cells. Now, you should know what is their role. Now, B cells are involved with humoral immunity, okay, where these B cells will release antibodies or immunoglobulins, they're also known as. So these immunoglobulins basically work against the pathogens that are present in the circulation, like the bacteria that is present in the circulation, okay. And they can go and bind to the pathogen, the antigen, and then they can help in removing that bacteria, pathogen, or antigen, whatever you can say, okay. Now what about the T cells? Now T cells are specific against various virus infected cell, or it can be a tumor cell, okay. So basically, T cells are specific for the cells that are infected, okay, or a tumor cell, okay. So T cells do not release antibodies, okay. What you can see over here is, don't get confused, is not an antibody, it is a T cell receptor that is present on the surface of the T cells, okay. So that is what it makes the acquired immunity. You have the B cells and you have the T cells, right? Now further, we will see each of them in more details. So before that, we will discuss about the primary immune response and secondary immune response. Now when the pathogen enters the body for the first time, okay, the response that is generated, so that is known as the primary immune response, okay. So that time there is an antibody that is released, so it is known as IgM. So there are 5 different types of antibodies. Okay, so the antibody over here is IgM. Now suppose again the pathogen enters for the second time. Now when it enters for the second time, the immune response that is generated, so now we call it as the secondary immune response. Okay, so primary is when the pathogen enters first time, secondary is when the pathogen enters for the second time, the same pathogen. Now if you compare the primary immune response, the if I talk about the concentration of antibody, so antibody is less. Okay, and it has a long lag period, around 5 to 7 days it takes. But if it is a secondary immune response, you can see the lag is very less, maybe around 3 days or 2 days, 2 to 3 we can say. And it peaks up also very early. Okay, like here it peaks in 14 days, here it will take less days, and the level of antibodies is much higher. So that is why the secondary immune response, when it gets activated, it will quickly try to control the pathogen. Okay, and the antibody that is responsive over here is known as IgG. Okay, so now what is humoral response and what is cellular response? So humoral response means, as I said, the B cells get activated. So when B cells get activated, they release antibodies. So if it's follow this figure, so a foreign body or a pathogen has entered, so it has been taken up by the macrophages. Okay, and then macrophages, what they do, they take the antigen, they process it. Okay, they break it down and then they present it. Okay, so they have certain surface markers, like it is known as MHC. So they present it to the T cells. Now, when they present to T cells will release certain cytokines. So these cytokines will activate the B cells. Now the B cells will release the antibodies. So what we see, what is humoral response? Humoral response is when the antibodies are released by the B cells. So the antibodies is coming under the humoral response, the function of the antibodies. Okay. Then what is cellular response? Cellular response, by the name itself, means cells are involved. Okay. So here, like T cells get activated, further they release cytokines. Then T cell is of two types. Okay. One is known as the T helper. So that this is, or the helper T cell you can call. And the other one is known as TC cell or cytotoxic T cell. Now, initially, the T helper gets activated. So this T helper basically releases cytokines, and those cytokines will activate the cytotoxic T cell. Okay, now when cytotoxic T cells become active, so they are capable of destroying virally infected cells. So here the cells are getting activated and they are giving a response. So we call it as the cellular response. Okay, so that is the difference between humoral response and cellular response. So humoral response will involve the B cells that release the antibodies. Cellular response will involve the T cells, okay, that get activated, and in turn they help in killing the viral infected cells. Okay, now this adaptive immunity further can be classified. Okay, you can call it as naturally acquired or artificially acquired, this adaptive immunity. Okay, now in that naturally acquired, you can also call it as active or passive. Similarly, artificially acquired can also be active or passive, or you can say this active immunity. Okay, so this active immunity can be natural, right? Other way around also, you can say the active immunity can be natural or it can be artificial, right? Similarly, the passive immunity can be natural, okay, or it can be artificial. Now what is this active naturally acquired adaptive immunity? So in this active naturally acquired adaptive immunity, here the antigens will enter the body. So it is active and it is naturally acquired, means when we have some infection, the antigen is entering the body and the B cells will get activated, they'll release antibodies. Okay, so in this way, this is known as active naturally acquired. So this is basically when you are suffering with some infection. Then when it will be a passive naturally acquired adaptive immunity, when directly antibodies will pass, okay, from the mother to the child's milk, right? So that is known as passive naturally acquired. Okay, so it is a passive immunity, but it is naturally acquired. So why passive? Because directly the antibodies are entering. Okay, the antigen is not inducing the release of antibody over here, right? Okay, then Active artificially acquired means here the antigens are introduced artificially, right? So you are injecting the antigens over here like in form of vaccines. So when vaccines enter the body, so it is active artificially acquired. So the vaccination part, so the vaccines is what? It is a weak form of the antigen that is given. So that antigen, when it enters, it will start, activating the B cells, T cells, and that is how we get the active artificially acquired adaptive immunity, right? Then the passive artificially acquired adaptive immunity is again some preformed antibodies will be injected into the serum. Okay, but here there is, there is the plasma therapy you might have heard nowadays that goes on. So here the antibodies, like whoever immune system is weak, suffering with some infection, so directly the antibodies can be passed from a healthy individual, or monoclonal antibodies can be given. So in that way it is known as passive artificially acquired adaptive immunity, right? Now next, what is antibody? So the structure of antibody is very important. Okay, so it is a glycoprotein. Okay, so glycoprotein means it has protein. So you can see there are four polypeptides. So this is 1, 2, 3, 4, and these two are known as light chains. Okay, so these two are the light chains, and these two are known as the heavy chains. Okay, so heavy chain and light chain is based on their molecular weight. Now they are proteins. Okay, so they'll have a amino terminal and they'll have a carboxyl terminal. Okay, so this side is the amino terminal and the other side is the carboxyl terminal. Okay, now at the amino terminal end, so this part of the antibody, okay, then at the amino terminal end, so this part is known as variable region. So this is variable region of light chain, this is variable region of heavy chain. Again, this is variable region of heavy chain and this is variable region of light chain. Okay, then the other part is known as the constant region. Okay, so this is constant region. So this part is constant region on heavy chain and this part is constant region on light chain. If you see over here, these polypeptides, so how they are, bound to each other. So basically there are disulfide bridges that hold them together. Now apart from interdisulfide linkages or bridges, so inter means one chain with another, there are also intradisulfide linkages, means within the same chain also you can see that there are intradisulfide linkages. Okay, so this structure is very important from your exam point of view. Sometimes they even ask how many disulfide linkages are present. Okay, so that is also important that you should know. Then you have to know about where is the amino terminal. Now the site where is the amino terminal, where we are calling it as the variable region. So these are the sites where the antigen binding sites are present. So this is where the antigen will come and bind. Okay, not to the other end but to this end, to the variable region, the antibody will come and the antigen will come and bind. Okay, means the antibody can bind to antigen at the variable region, right? Now this antibody that you can see over here, okay, so it has no the why we call it as variable, because this part, okay, where the amino acids are present, so they're highly variable in one amino acid to another, one antibody compared to another antibody. So the amino acids are variable, means the same amino acid is not present in another antibody, so the amino acids vary over here. So that is how each antibody is specific for a particular type of antigen, right? So they are specific because their variable region is different in different antibodies, while constant region will have some very similar type of amino acid that is present, right? So this is about the structure of antibody. Then this, if you talk about the innate and adaptive immunity, so what all comes under the cellular components and the humoral components. Now for cellular components, in innate immunity you'll include the various cells that we discussed, like monocytes, macrophages, neutrophils, natural killer cells, mast cells. For the humoral component, I told you for humoral it is the antibodies, right? But if they ask innate immunity humoral component, humoral itself means soluble components. If they ask about innate immunity humoral components, so it will be the proteins that are involved. Okay, those soluble components like complement proteins are there, acute phase proteins. So one of the acute phase proteins is C-reactive protein, then cytokines, interferon, I told you, right? So these will come under the humoral components of innate immunity. Now, what are the humoral components of adaptive immunity? It is the immunoglobulins or the antibodies, right? And then what is the cellular component for adaptive immunity? The T cells and the B cells, or T lymphocytes and B lymphocytes. And as I said, majorly they are of two types: T cells or helper T cells, or cytotoxic T cells. Okay, now if you see these helper T cells, they have they have a T cell receptor that is present, and along with it they have a molecule. Okay, like if it is a T helper cell, so it will have CD4 surface marker. If it is a TC cell, or cytotoxic T cell that we call, so they also have this T cell receptor. Now along with that they have a CD8 molecule, right? So this table is again important from your exam point of view. Now apart from that, now we'll see the B cells and T cells. What are the surface markers that are present on the B cells? Because they are very important. These B cells, they release the antibodies, right? So the first thing that you should know about the B cells is, where do they develop? So development of B cells starts in your bone marrow, and they mature also in bone marrow, okay, in mammals. But if it is a bird, so their site of maturation is another organ that is known as bursa fibriscus. Okay, and each B cell is programmed to express an antibody for a particular antigen. So as I said, each B cell, if it is a B1 cell, it will release one antibody type, that is antibody 1. So it is specific for only one type of antigen. So each B cell is specific. If it is a B2, it will release some other antibody, and it will be specific for some other antigen. Okay, so in this way there are different B cells, there are millions of B cells, and they are specific for different antigens. Okay, so they display on the surface. If you see, if it is a B cell, so on their surface what they have a receptor. So there is known as B cell receptor. Now what is this B cell receptor? This is again antibody that is present. Okay, it is an antibody. Basically the antibody that can be present is IgM as well as IgD on the surface of the B cell, while other antibodies, they are released. Okay, then apart from that, it has a CD45 as a marker. Then it can have MHC class 2. Okay, MHC class 2, why it is present? Basically because B cells can take the antigen, they can process it, and they can present it on their MHC class 2. And when they present it on their MHC class 2, they activate the T cells. Okay, so they have MHC class 2. Then they have certain receptors that are present, that is for the complement proteins. So CR1, CR2 are the various receptors for the complement proteins. Then it has another molecule that is known as B7. Okay, so B7 will interact with T cell. I told you, right? They have MHC, they activate the T cells. Now T cells will have CD28 and B cells will have B7. So in this way the B7 and CD28 interact. Okay, then they have another molecule that is CD40 also. CD40 also helps when the B cells have to be activated. So the CD40 on B cell and CD40L on the T cell need to interact. Okay, then majorly all the B cells will have CD19 surface marker, except for the plasma cells and another type of cells, that is follicular dendritic cells. Okay, apart from that, all B cells will express this marker, that is your CD19. Now let's talk about the T lymphocytes. Okay, so T lymphocytes, their development also starts in bone marrow. Okay, but remember they mature in the thymus. So that is how they get their name, thymocytes or T lymphocytes. The T is because they mature in the thymus. Okay, so you can see this is a T helper cell they have shown over here. So what all will be present on a T cell? Okay, so first thing, as I said, T cells will have T cell receptor, and depending on what type of cell it is, it can either have CD4 or it can have CD8. Okay, it is a co-receptor, you can say. Then they have CD28 to interact with B7, I told you, right? And then they can also have CD40L. Okay, now if you see this figure over here, So it is a T cell. Okay, so T helper cell means it will express CD4. Okay, so remember T helper cells will have CD4. CD8 will be for TC cells or cytotoxic T cells. And they also have another marker, that is CD45 for signal transduction. Okay. Now T cells, they cannot identify the antigen as it is, like the B cells. Okay. So for T cell to identify the antigen has to be taken up. Like here, macrophage will take the antigen, it will process it, it will break it down, and then it will present on its MHC class 2. So when it present in MHC class 2, it will activate the T cells, right? So further, it needs an interaction between B7 and CD28, as you can see over here, right? So both these are required to activate the T cell, both this signaling. So when the T cells get activated, they will release the cytokines. Okay. Now, the next figure that you can see over here is, it is a B cell. So B cell can also take the antigen, they can process it, and they also have MHC class 2, they can also present it. Okay, and this time they have presented to the T helper cells. Now, to activate the B cells, now suppose the T helper cell got activated, okay, by these two interactions. First is the interaction with the antigen, and second is between B7 and CD28. So these T cells will release the cytokines. Now, when cytokines are released, the B cells will get activated. Okay, and to activate the B cell, this signal is also important. Okay, first thing, it has to take the antigen. Okay, so that will give the first signal. Second is the interaction between CD40 and CD40L that is present on T cells. Okay, so this will give the second signal. After that, only the B cells will get activated, and once they get activated, further they will differentiate and they will start releasing the antibodies. Okay, so I'm sure everybody is understanding. If anyone has any query, they can ask me. Okay. Okay, so let's continue now. So that was about the immune cells. Okay, next, so already we discussed about the immunity. Okay, now we are going to discuss about the immunization part. Okay, so it can be either active or passive, right? So passive is when preformed antibodies are given, but this type of preformed antibodies, when they are given, when they are transferred to a sick person, so the immunity that it provides, the protection that it provides, is just for a transient time, it is for a very short duration. Okay, so what are the various examples? Like maternal antibodies passes to the fetus through the placenta. Okay, as well as antibody therapy when it is given. Okay, or when the person is suffering with some immunodeficiency diseases, then also certain specific antibodies are given, right? So all this is about the passive immunization. Now, what is active immunization? Active immunization means, here, the person will come in contact with the foreign substance or the antigen, but the antigen is weak over here, so it is given in the form of vaccines. So the active immunization is a long term protection where the antigen is given, not the antibody. Okay, but it provides long term protection, it activates the memory cells, and here there is actual exposure with the antigen, right? Now, so now we will discuss about the topic, that is vaccines. Now, for preparing any kind of vaccines, two things are very important. Okay, one thing is, the antigen, the vaccine is something like you are giving the antigen, right? So this antigen, when it enters the body, so first thing, it should give rise to immune response. Okay, and second thing is, it should give rise to memory cells. What is the point if the memory cells are not getting activated? Okay, then the whole concept of vaccines will be a failure, right? So these two responses or these two requirements have to be met, only then it can be a successful vaccine, right? Okay, so What are the different types of vaccines? Though there are few more types of vaccines, but these are the most important ones that I have listed from your exam point of view. So do remember their examples, what they are, okay, and what type of immune response it gives, whether it activates the B cells or whether it activates the T cells. So that also you should know. Okay. Now, so it can be a whole organism vaccine. So we'll see each of them, okay, or it can be certain toxoids. Then there are polysaccharide and polypeptide vaccines. Then there are recombinant vaccines. So first we'll talk about this attenuated or live vaccines. Okay, attenuated means the antigen is weakened over here. Okay, the pathogen, you are weakening the pathogen and then you are injecting it as a vaccine. Okay, so here suppose there is, how will you weaken or how will you attenuate a particular pathogen? Okay, so the pathogen will lose the ability to cause significant disease. So how it is done basically, suppose a bacteria, no, you know that bacteria grow in certain specific medium, right? You have LB media and there are specific different types of media for different strains of bacteria. Now what you do is to attenuate the bacteria, you grow it in an adverse condition. Okay, instead of the lurea bertini media, you are providing it, suppose bile. Okay, so it is a completely different medium. So now those bacteria will not survive, right? Now majority of bacteria will not survive, but some of them will survive, right? So those that survive, those are known as your attenuated strains, okay, or attenuated, it will act as attenuated vaccine, because those bacteria, again, when you inject it into a human host where the conditions are suitable, so they cannot cause infection. Okay, so this is about the live attenuated vaccines. So a virus must be grown in cell type that is not normal host. So for bacteria, you have to grow them in a different medium that is not suitable. For a virus, you have to grow it in cell type that is not a normal host. So the virus will again start accumulating certain mutations. So those mutations will again weaken the virus and then it can be used as an attenuated vaccine. So the examples over here is for measles, mumps, the Sabin polio vaccine. So all these examples comes under the attenuated type of vaccines where the antigen is live but it cannot cause the significant disease. Next, inactivated or killed vaccines. So these both types are your whole organisms where they are being used, but either it can be weakened or it can be killed. Okay. Now killing the pathogen can be done either by giving certain heat treatments, okay, or certain chemical treatments. So the pathogen cannot replicate in normal host. Okay. But when you are doing that, okay, when you're killing it, so you have to make sure that the epitopes, okay, epitopes are that part of the pathogen that actually bind to the uh, antibodies, right? So those epitopes have to be maintained. Okay, the epitope should not be destroyed, only then it can be used as a vaccine. Now this type of vaccination will require boosters. Why? Because the pathogen is killed. Okay, so it cannot, it cannot keep on generating the memory cells. So because it is, the pathogen is killed, so it is basically the effector response get activated, but the memory response is not getting activated. So that is why often boosters are required. Okay, and another disadvantage is because here the pathogen has to be grown in large number. So again, it is a risk for the people who are involved in this kind of vaccine production. And there's also a possibility that some of the pathogen might not be killed also. And then it might survive. And then again, when it is used as a vaccine, it can cause the disease. Okay, so these are some of the risks that are associated with this killed type of vaccines, right? But another important point over here is it provides humoral immunity. Why humoral immunity? Basically the B cells will get activated over here. Okay, so the examples are Salk vaccine, okay, then influenza vaccine, pertussis vaccine. So Salk vaccine was the vaccine that was made against for the polio. Okay, so Salk and Sabin, two types were made, and this Salk, there was chances of failure of inactivation also. Okay, so certain failures were also observed with this vaccine. Next, coming to the toxoid vaccine. Now, by the term toxoid vaccine means now various bacteria they release toxins. Okay, like diphtheria, tetanus. So those toxins, or you can say the exotoxins, that are being produced. So you can modify those toxins and you can make it non-toxic. Okay, but though that non-toxic bacteria, the non-toxic toxin part, okay, will have the capacity to lead to the formation of antitoxins. Okay, so understand what you're doing over here. You take the bacterial toxin, okay, you modify it so that the the epitopes are still maintained, okay, but the toxin is not very, it is not harmful. And then when it is injected into the individual, so it can activate the immune response of that individual and it can give rise to antitoxins. So these are like your antibodies against the toxin, so we call it as antitoxins. Okay, so you need to purify the bacterial exotoxin, you need to inactivate the toxin. So by using various chemicals like formaldehyde can be used, okay, and other chemicals are also available by which you can make it, you can make it non-toxic, right, by inactivating the toxin. And then when it is injected into the individual, so it will give rise to anti-toxoid antibodies. So these antibodies will bind to the toxin if a toxin is entered, and then it will neutralize the toxin. Okay, so antibodies bind to the toxin, and this is known as your neutralization. But again, over here, what is the requirement? Epitope should not be altered. If the epitope of a particular toxin is altered, okay, then it again becomes difficult. Okay, it cannot act as a vaccine, it cannot activate the immune response where the antitoxoid antibodies might not be released. Okay, so examples of toxoid vaccines, what are those? Tetanus toxoid and diphtheria toxoid. Okay, so these, like, tetanus is caused by the bacteria, okay, and diphtheria, these are bacterial toxins. So for them, for these toxoid, you have this toxoid kind of vaccines. Okay, next type of vaccine is recombinant vector vaccines. Now, recombinant vector vaccines, by the term itself, means recombinant DNA technology will be used over here. Okay, now in recombinant DNA technology, what is being done is, so the gene, okay, is inserted into a plasmid. So this gene will encode certain, virulent antigens. Okay, so maybe a pathogen has some virulent antigen, so you take the gene from that particular pathogen and then insert it into the plasmid, and then you inject this into the individual, into the human host, okay, into the host cells. So now when this gene of interest enters into the host cells, it will start making those virulent antigens. Okay, so here you are not injecting the pathogen, but you are only allowing the gene of interest to be expressed in the cells. So when that gene of interest get expressed, so now what will happen? It will start making that particular antigen, okay, maybe a polysaccharide. Okay, means basically this is for the proteins, okay, because the genes get expressed and they start forming the protein. So the antigen over here that is being formed is for proteins. Okay, so the gene encoding virulent antigens of the various virulent pathogens will be introduced in this attenuated viruses or bacteria, and the attenuated organism will act as a vector. Okay, so the vector over here, it can be a virus or even a bacteria, and this whole, combination of it, the whole assembly is used as a vaccine. So one of the most successful recombinant vector vaccines is the hepatitis B vaccine. Okay, where the hepatitis surface antigen that is encoded by the gene, so that particular gene that encodes the hepatitis surface antigen is injected. Okay, and the vector is created and then it is allowed for expression. Okay, next is the polysaccharide type of vaccines. So polysaccharide vaccines are those where, the polysaccharide capsules of the bacterias, so they are used as a antigen over here. Okay, only the capsule or the sugar molecule, you can say. Okay, because the polysaccharides are the sugars, so only the sugars are used over here as an antigen. So here basically hydrophilic polysaccharide capsules of various pathogens are used, like streptococcus, okay, Neisseria meningitis. So this coating of the capsule with antibodies, or it can be also these capsules can be coated with complement proteins. So when they are coated with these kind of antibodies and complement proteins, so their size increases, okay, and that is how phagocytosis will get enhanced. So the macrophages, neutrophils, they can phagocytose these type of pathogens. Okay, where you have the capsule and if it is coated with antibodies, so it will be a greater enhancement. So when it is taken up by these pathogens, by these phagocytic cells, so they'll activate the B cells over here. Okay, and IgM antibody is released, but there is no class switching. Class switching means from IgM it cannot change to any other kind of antibody, whether IgG or IgA. Okay, so there is no class switching over here. So this will activate the B cells. So majorly the humoral response get activated. You have to remember what type of response? Humoral. It cannot activate the T helper cells. Means the cell mediated immune system cannot get activated. Okay, because it is only the sugar molecules that are being used. But yes, if you conjugate this polysaccharide to a particular protein, okay, so we call it as a conjugated vaccine. Okay, so when you conjugate it with the carrier protein, so their size increases, and when their size increases, so that time they can also activate the B cells as well as the T cells. Okay, so those are known as your conjugated vaccines. So now for this polysaccharide vaccines, what are the various examples? Like you have streptococcus pneumoniae is one example, vaccine against the streptococcus pneumoniae, and Neisseria meningitis, right? So these examples are very important. So that is why I am sharing a table with you all. So this is important from your exam point of view. So it has a classification of the various common vaccines, like which are inactivated vaccines like anthrax, cholera, pertussis, plague. Okay, hepatitis. Now if you talk about viral particles like hepatitis A, influenza, okay, polio, salk, rabies, against rabies, rubella. So all these are inactivated type of vaccines. Then which is live attenuated, where for tuberculosis is used, for typhoid. So these are for bacterial cells. For viral particles, for measles, mumps, and polio, sabin. Okay, then a chicken pox that is caused by varicella zoster. So that also is your live attenuated. And there is another disease that is known as yellow fever. Okay, so this is by the virus. So this also is an example of live attenuated. Then for toxoids, as we saw, diphtheria and tetanus are the examples. For the capsular polysaccharides, we have Haemophilus influenza type B, where the polysaccharide is conjugated with protein carrier. Then only polysaccharide is for Neisseria meningitis as well as Streptococcus pneumoniae, where there are 23 distinct capsular polysaccharides. So all of this has been studied and it has been shown that it is capable of acting as a vaccine. Okay. Now, apart from that, the recombinant surface antigen, that is the example is hepatitis B, where the type of vaccine it is recombinant surface antigen, where the gene that encodes the surface antigen for this hepatitis is being expressed. Okay, so with that we come to the end of our session. And now we'll try to solve few questions and let's see how much you can remember. Okay, and meanwhile, if anybody has any queries, they can ask me. So which of the following cells is a part of immune system? Anybody can answer this quickly? Macrophage, eosinophils, lymphocytes, all of the above. Which of the following cells is a part of the immune system? So it is a simple question. Anybody knows the answer of this? What should be? All of the above. Very good, Aparna. Right. So as we saw, okay, all these cells are involved. Like macrophages, we saw it can perform phagocytosis. So remember which cell. Okay, eosinophils also. Basically, eosinophils, they are released when there is an allergic condition. Okay, when you're suffering with allergy, you'll see the eosinophils levels goes high. Lymphocytes, yes, lymphocytes, if there is any kind of infection, so the B cells, T cells, so the adaptive immunity get activated. So the answer is all of the above. Correct, Aparna. Now next, what are antibodies? Are they DNAs and RNAs, carbohydrates, glycoproteins, all of the above? Is it a carbohydrate? Antibodies? Glycoprotein, correct. Yes, right, Aparna. So the right answer over here is glycoprotein. Okay, though it is glycosylated. Okay, glycosylated means it will have certain sugars. Okay, but you will not say it is a carbohydrate, it is a protein. Okay, as we saw, there are two light chains and there are two heavy chains. So the right answer is your option 3, correct. Okay, then we'll see one more question. And then, yes, light chain and heavy chain of antibody are joined by, so you have to remember the structure of antibodies properly. So whether they are joined by hydrogen bond, hydrophobic bond, ionic bond, or disulfide bond. So what type of bonds are responsible for joining the light chain and heavy chain? Okay, Aparna. So it is a disulfide bond, correct. Okay, so already we saw in the figure, so you have to remember also, okay, what it is. It is a disulfide bond, so you need to know the structure of the antibody thoroughly over here. So the right answer is your option 4. Good, everyone. So now with this, I would come to the end of the session. So before that, now if you want any further help, so please be in touch with BioTecNika on Telegram. So the link is there. You can come and join this link and do download the BioTecNika app, right? So for any kind of classes, podcast, right, webinars, newsletters, so you will get information about all of these that are conducted for you all. So you should join over here so that you be in touch with BioTecNika and you can avail these facilities. And if you have any queries related to any technical queries, any classes that you want to join, anything you feel you need to contact BioTecNika, so do email at info@biotecnica.org. You can even contact our toll-free number, that is 1800 or you can call at 08050997000. And we would love to hear from you. So do post your feedback, your testimonials about the class, how it is helping to enhance your knowledge. That is very important. So you can share with us and you can write us at zipmessage.com/biotecnica. Thank you so much for being an attentive audience and listening and understanding carefully. So before I end, I would say this: success is the sum of small efforts repeated day in and day out, as told by Robert Collier. So do follow it. Okay, the exams are nearby. You have the CUET UG exam very nearby. So prepare well for the exam, and I wish you all the very best. Thank you, everyone.